Hi, my name is Eric Siniseros. I'm in the Mechanical Engineering Department, working on my master's thesis. Today I'll be talking to you about us measuring forces with biocompatible photoelastic materials. This research is in collaboration with my advisor, Henry Fu, in mechanical engineering, and Zephyr McCormick in physics. On my title slide here, you can see some photoelastic spheres that we've made. On the right side, I'm pushing on them with a pair of tweezers, and it makes the force change that propagate through this granular media visible in the form of brighter light right here. So the purpose and the goals of this research is to create a system where we can measure organisms in a more natural environment, I guess. This instrumentation must work in environments with complex geometries, and it must be able to measure forces dynamically and on a small scale, hopefully a micro scale, by the time we're done. So to start, I'd like to give you some background on photoelasticity. This is a picture of a wrench, a photoelastic wrench turning a hexagonal bolt on the left side. You can see the visualization of the stress propagate, or the high stress concentration areas here along this back edge, and here where the bolt's making contact with the wrench. The wrench is made of a birefringent material, and so the speed of light is different through different materials. A birefringent material has a speed of light different in itself in different axes of propagation. So here you can see a polarized light source coming into the top of this birefringent material. It then propagates at different angles at the interface, taking different path lengths and speeds through the material, and then essentially recombining down here to give you a relative retardation of one propagation to the other. That's about what I just covered. A very basic equation to help you guys get your heads around this is this one here. Sigma 1 and 2 are the principal stresses. N is proportional to retardation, which is what we're measuring in our experiments. F sub sigma is the fringe value, which is determined through a calibration. And H is the path length, not the path length on the previous side, but the thickness of the material. So a basic instrument to measure this photoelastic effect is called a polariscope. It consists of a light source, first polarizer, notice it's oriented vertically, quarter wave plate, which is an optional addition just to change what type of polarized light you're measuring. You then have your birefringent specimen in the middle with the first and second principal strain axes. Another quarter wave plate. If you have the first one, you have to have the second one. And most importantly, your analyzer, which is oriented 90 degrees to the first. So the light source passes through the first polarizer, filtering out all light that's not polarized in the vertical direction. Then if you skip down here to the analyzer, this one is oriented 90 degrees to it, which would block all light in an unloaded specimen. When the specimen's loaded and it shifts the polarization, you can see the difference in those two polarization paths with your camera down here. So here's another image. This is a washer. Photoelastic washer, black and white this time, being loaded axially at the top and the bottom. Again, you can see the stress concentrations here and here. What you need to pay attention to are these black lines. This is just a monochromatic version of that colorful wrench we saw earlier. Each black line is called, is where that F sub sigma value is a whole number, meaning 180 degrees of relative retardation of that light, so it's all blocked out. You can say the same thing about the white lines, just they're 180 degrees out of phase with the black lines. And the way you can determine forces in this is counting these black lines, which have a fringe number, and then if you know your elastic modulus of your material, you can calculate the force. So this is an experiment I ran, just to sum this up. These are unloaded photoelastic spheres, and this is a photoelastic sphere that I'm barely pinching with gelatin, and you can see it's brighter. To make these spheres, we are using gelatin, which acts like a long-chain polymer, sort of like polypropylene, and it is photoelastic. We load the liquid gelatin, it's the same stuff that's in Jell-O, into a syringe, and then we drop it into a nonpolar solvent, so we get the oil and water effect, except the gelatin will sink through it. We use toluene. And we allow the surface tension of that gelatin to shape itself into a sphere on its way down while it cools. This allows for excellent consistency and repeatability in our spheres. We can also do this with a burette and set it up automatically. We also have a method that we're working on to use micro-scale capillary tubes to make spheres about 100 microns in diameter. Right now we're just controlling the size with different dispensing tips on the syringe. We can go from about a millimeter, about a half a millimeter up to about 5 millimeters. So you can see liquid gelatin in our beaker on the hot plate, and here's our graduated cylinder of liquid cooled toluene. This is being cooled with dry ice. We just use water ice now, and it just gets dropped right in the top here. Here's an example of the size variation of spheres we can make. We can make them all homogeneous in size, or we can make a huge variation in size like this. The small ones in here are about 200 microns, and the larger ones, like here, about 5 millimeters. During this manufacturing process, just like anything when you're making, you end up with a residual stress problem. This shows a signal in unloaded spheres, which is undesirable, so those dark spheres will be bright. They also dehydrate over time, changing their size and causing residual stress. We tried storing the spheres in pure water to keep them hydrated, but they ended up just ballooning. I'll explain that a little bit more in a minute. We also tried toluene as a storage medium, but it did not stop the dehydration either. Usually the residual stress problem is solved with annealing, which means heating the material almost to its melting point and then cooling it back down, but the gelatin will just melt if we do that. So here's an example of this residual stress. These are the dark spheres. That's what we want. These are spheres in a hypertonic state, meaning they swelled with water. These are spheres in a hypotonic state. The sphere is going to be like this, and there's going to be a plus pattern in the middle, meaning they have dehydrated and shrunk. So we came up with a solution to this, storing the spheres in an ionic solution. The inspiration for this was cell biology. Cells have a semi-permeable membrane, and fluid exchange, osmotic fluid exchange across the cell membrane happens based on ionic balance. I know there's some biology people here, so please don't grill me on that. That's not my field at all. This can be basically understood by the osmotic pressure in the Morse equation, where pi is the osmotic pressure, the pressure gradient across that membrane. Van Hoff factor, which is just a non-dimensional number, the molarity of our ionic solution, the gas constant, and our absolute pressure. The gelatin inside the sphere acts like one ion because there's loose ends on the protein chain, and then we have a saline solution on the other side of that membrane to facilitate that fluid exchange. And this equation shows that the molar concentration of saline is linear with the concentration of gelatin in our spheres. And we verified that experimentally. Our gelatin calibration apparatus is this fluoroscope here, which I made. It has a monochromatic green light source. Here's our first linear or circular polarizer. We have a high precision balance with a vertical displacement gauge, so we're measuring the reaction force at the bottom of the spheres, and the sphere is placed right here. We then have our second set of polarizers oriented 90 degrees to the first, and finally just a Canon digital camera to capture the image here. Here is an image of the sphere being repeatedly loaded, and you can see as it's increasing in load, it gets brighter, which is exactly what we want and expected. We then do an image analysis on the pictures with MATLAB. We convert the image to RGB which just gives a numeric value to the red, green, and blue ratios in the pixel. We then subtract the background image. We took an image without a sphere in the polariscope, and we subtract that from the sphere image. We then identify the circle using the image processing toolbox. Mask areas outside the circle, and then we simply sum the pixel values of our green light inside the circle to get our final number. Here's an initial response curve that we came up with. As we load the sphere, there's a dip at the beginning, which we have not explained yet. We're working on that. The response curve then becomes linear up to a point, and then maxes out. So our upper limit of measurement here is where the signal coming through the sphere has blown out the sensors in the CCD image, and that just reads as white light. So some other experiments we'd like to try in the future. I'm sorry, this is our application. So this analysis can be performed statically on a network of multiple spheres. We can do a dynamic analysis using a normal, high-def video camera or a monochromatic high-speed camera, which we also have. We can also easily fabricate other geometries besides spheres. So we could do a bacterial shape, spirochete shape, anything you can cast in a mold you can do with gelatin. We've recently come across some other research where they've made microscale filaments in a network. There's a lot of research in my lab with cervical mucus and how sperm and other organisms move through bodily fluids. And those fluids look like a network of tangled rubber bands. We're not going to be able to get to that scale, but hopefully we can create a network scaled up from that and still get relative results. So there's great interest in measuring forces generated by organisms without disrupting their natural environment. This goes for worms, tunneling worms, nematodes, which I'll talk about in a minute. There's been some work with plant root growth for soil erosion prevention, and also watershed studies through soil. And we would like to also be able to measure forces in a pressure field, which we'll be working on in the next month. So here is a picture of a nematode. It's 100 microns long and 10 microns in diameter. 100 microns is about the width of your hair. It has 6 motor neurons shown here, and it was the first organism to be completely DNA sequenced. It's very popular in neural networking and genetic modification research because it's very easy to cultivate. They also laser off these neurons so they can just study the ones they want, and they have not been able to determine the minimum force necessary to stimulate a neuron response so far. And our hopes is that our research will further that purpose. Most of the experiments on nematodes look like this. These are elastic PDMS pillars. They know the modulus of elasticity of these things, so they can monitor the deflection of them and then calculate how much force the worm is exerting on them. As you can see, the worm's kind of wedging itself between them, so it's possible that all these forces are not being generated by the worm itself, and it's more of a prying apart action. We think that a packed bed of microspheres would be a more realistic environment to measure forces in. So our future work, we'd like to experiment with glutaraldehyde fixing, which will cross-link the proteins in the gelatin. Same process that you use to preserve animals in biology lab. This makes them insoluble in water, much more elastic and much more resilient to damage. Another material we can use is sodium alginate, which is less toxic, so we could have applications in food safety. It's solidified by a chemical reaction, so it's not soluble at all. And we can make more complex properties because it's more stable. And we have succeeded in making small photoelastic filaments with these, so we can measure the tension force visually. So in conclusion, we talked about basic photoelastic concepts, how gelatin's a birefringent material. We talked about how we fabricate our gelatin spheres and using Morse equation and osmotic pressure to eliminate the residual strain. And we talked about some static and dynamic applications, as well as studies on nematodes and root systems that we'd like to explore in the future. I'll leave that up for you guys. Any questions?
We have time for one question.
I have a question. Can you functionalize the surface of your spheres with other proteins or cross linkers, anything like that? Anything that will make it interact with other molecules?
I have no idea. That was a short question. One more?
Why do you use toluene?
Toluene is it's available through the chem store. It's cheap,
Okay.
and it's not soluble in water. So we end up with that water and oil emulsion action that gets our spheres.
Oil.
We tried vegetable oil. It has low viscosity too, which is important.
Okay.
We tried using vegetable oil, and the viscosity was too high. And even though our spheres took a couple minutes to sink to the bottom, they were not solidified by the time they got to the bottom. In toluene, it's maybe a second or two. And we figured that's because the Reynolds number of that type of flow would be really small, and there would be almost no convective heat transfer around the sphere. So it would just hold all its heat in. Even maybe take the heated oil with it as it goes to the bottom. That's all. All right. Thank you.